And I just wanted to first of all thank you all for coming to the first of our 4 invited speakers for the Bioinformatics and Computational Biology seminar series. Tonight we are honored to have a speaker who is a leader in the field of computational biomodeling and systems biology. I met this speaker when I participated in an REU at Virginia Tech on modeling and simulation in systems biology, which he directed. And it was this program that led me to go on to pursue graduate studies in bioinformatics and computational biology. So when I was asked to find a speaker who could introduce the Iowa State University community to systems biology, I couldn't think of a better person than the one who introduced me. So his professional preparation includes a bachelor's degree from the University of Munich in Germany, a master's degree at Indiana University, and a PhD at Northwestern University in the field of mathematics. So since 2001, he's been a professor at the Virginia Bioinformatics Institute and a professor in the math department at Virginia Tech. In addition to being an adjunct professor at, in the Department of Cancer Biology at Wake Forest, an affiliate faculty member at the Virginia Tech Wake Forest University School of Biomedical Engineering and Sciences. In addition to all these faculty positions, he is also the Director of Education and Outreach at the Virginia Biotech Bioinformatics Institute. In 2010, this is really interesting, he was invited by members of the U.S. House Committee on Science and Technology's Research and Science Education Subcommittee to testify on some of the research challenges that are encountered at the interface of biology and mathematics. Some of his research includes network inference and parameter estimation for discrete molecular species interaction models. Recently, he's been working on with researchers at Wake Forest University to investigate intracellular iron metabolism and its relationship with breast cancer. So tonight, his talk will be on cancer systems biology. So I'm pleased to present Dr. Reinhard Laubenbacher.
Well, thank you for this wonderful introduction. Now I have to live up to it. And I guess I'm the first speaker in this series, so I better not screw up, otherwise these people are not going to show up the next time. So I put a very generic title up there, but in fact I'm going to mostly talk about something quite concrete, which is the work that Ali mentioned on breast cancer and iron metabolism. And so I think I have 45 minutes roughly, is that right? Okay. Very good, so feel free to interrupt in any time in between. So maybe the first thing I would like to do is, so many people talk about systems biology and most people mean different things by them, and it's probably not so useful to, give the definition, but I'd like to give you what I view as systems biology and how it relates to bioinformatics, right? And that relationship is sometimes maybe not so clear, and I'll give you my version of it. Anybody else who would stand here will give you a totally different version, so you will need to. And so I will refer to a higher authority. In this, Hans Westerhoff, a very eminent systems biologist at the Free University in Amsterdam. And so here's how he and his co-authors describe systems biology in a 2006 article. The advent of functional genomics has enabled the molecular biosciences to come a long way towards characterizing the molecular constituents of life, right? So those are the genes, proteins, metabolites, lipids, whatever, all the stuff that swishes around in cells. And yet the challenge for biology overall is to understand how organisms function. Okay, so maybe not all biologists might agree with this, but That's in any case Westorff's view. So by discovering how function arises in dynamic interactions between these molecular constituents, right, systems biology addresses the missing links between molecules and physiology. Okay, so genotype, phenotype, the connection. And for me, the operative term here is dynamic interactions, right? I think that ultimately the key is in understanding the dynamics of these systems. And many times, we draw static pictures, right? Most of the pictures we draw, whether they're protein-protein interaction networks or metabolic pathways, you go to KEGG, and what you see, of course, is static pictures. And that's important to have, but it cannot be the final step, right? It needs to go one step further, and that's what my research group has focused on mostly, is looking at dynamic interaction, whether it is the inference of dynamic regulatory networks from data, or whether it's metabolic networks. So how does this relate, in my mind, to bioinformatics? Right? I think bioinformatics is a part of systems biology. Right? If so I see a systems biology approach as quote unquote unbiased approach. You come to a system, whatever that system may be. It might be the entire metabolic network in E. coli or something like that, right? You come to it, you measure whatever you can measure about this network. You measure all the metabolites, you measure all the mRNAs. And then you use bioinformatics to tell you what is important for the particular problem, for the particular condition, or the particular experiment that you're interested in. Right? You can do that by a number of different methodologies. You might look at differentially expressed genes in microarrays. You might look at changing metabolites in mass spectra, whatever you do. And then once you have pulled out what's of interest, say, all the transcription factors you're interested in that actually do something in the particular cell type you're interested in, and once you know which transcription factors regulate which others, then you can begin to study these dynamic interactions. So bioinformatics is sort of step one of a systems biology project. That's how I personally see this. Other people might disagree. They might say if you do a genome-wide association study, then that is a systems biology project, right? Which is okay. Right? I once was at a systems biology conference where a speaker tried to actually make sort of a binding definition of what systems biology is, which ended up in complete chaos. So I wouldn't do that, but anyway, so that's my view. And let me just show you an image from a review paper we wrote a little while ago that illustrates this. What you have here on the left side is you have the kind of diagram you find in many molecular biology papers. Right? It's maybe a phenomenological kind of network. So this is about the AKT, action of AKT. It's an interesting kinase that plays a big role in various phenomena having to do with cancer. And you can see sort of what AKT affects. Those are proteins. Those are processes like glycolysis, and you see here other proteins that regulate AKT. And so you might assemble this from partly from the literature, partly from experiments you've done, maybe knockout experiments or whatever. So this is one level of knowledge, and it's probably where a large part of our knowledge resides, is at that level. Okay. Now, if you look at this middle picture, right, so this is a picture that's made with a software called Ingenuity Pathways Analysis, IPA, which I'm sure a lot of you know. Right, so one of the things you can do with IPA is you have a list of genes, for example, a list of genes you decided were differentially regulated in a particular experiment you did, and you put those into IPA, and IPA essentially gives you sort of all the different pathways that these genes are involved in the different connections, right? And the way IPA does this is it curates the literature, and so it gives you a view of what is known in the literature about these particular genes. And so what you see here is, right, here's AKT, but of course AKT doesn't live in isolation. It has to do with the COX-2 pathway. It is involved with NF-kappa B. So you can see that really what you need to look at maybe is this whole spaghetti picture. But of course, as spaghetti pictures go, they don't tell you all that much. Right, which is, for example, this one doesn't tell you all that much, right? What you would like to know is what happens if I modify this particular protein. Let's say I knock down this particular protein, then how does this perturbation propagate through this network? So what happens at the other side of it, right? Then you cannot begin to understand from this picture. And So that's what this right panel starts to do. So what you see here is a little piece of a computer model, if you like, that's built using differential equations. And what it does is it represents actually sort of roughly this chunk of this picture, I think, if I remember correctly. And what it does is, so you write down differential equations for the different proteins and how they affect each other. And once you have that, right, then I can generate these concentration curves over time, right? And I can fiddle with one of these proteins in this system of differential equations, and I can see how this effect propagates as I look over time at how these concentrations change. And of course, these pictures, right, they're very close to what you do with your experimental data in your lab, right? You plot the data points and you connect them with lines. Right? And that's exactly more or less what this is. And so you can begin to study the dynamics of this network. Okay. And so I think, one of the big challenges we have ahead of us in the next however many years is to take a lot of what we know that exists at this level and lift it up to this level. Okay, now a lot of things are needed. So when you write down these differential equations, you will have lots and lots of parameters in these different kinds of, rate constants and other kinds of parameters, maybe diffusion coefficients. And in many cases, those are not known, right? And probably you will get nobody to measure them for you. So that's certainly a challenge. We'll need more data, right? It's the common wisdom is that biology is drowning in data. And that's true for some types of data. It is not true for other types of data. So for the types of data to go from here to here, we have a great need for more data. So if you go and look, for example, for time course data of gene expression in yeast, you can probably count them on the fingers of one hand if you look at really good time courses. So there's not so much in that respect. Okay, so what I want to do is I want to show you in terms of a particular example. It's going to be a very small example, but I want to show you exactly the progression through these three steps, and give you a little bit of an idea of what sorts of things you can do with a computer model at that level. So the case study I want to show you is iron metabolism in breast cancer. I'm very fortunate to have some great collaborators at Wake Forest University Medical Center at the Comprehensive Cancer Center who have studied iron metabolism in breast cancer for a long time. So I'll talk a little bit about this. So first of all, what does iron metabolism have to do with breast cancer? And here we're talking about intracellular iron metabolism, not systemic iron metabolism. So there are a number of things that are known about this. First of all, it's known that if you just measure the proteins that are involved in regulating iron homeostasis in normal breast epithelial cells and in malignant cells, you get quite different expression levels. Certainly one difference is that malignant cells require more iron because they grow much more quickly. And you need iron for many things. You build up DNA, you need it for all kinds of proteins and other things. And it's been observed in a number of places that individuals who accumulate excess iron have an increased risk of developing cancer. And in fact, it makes a difference in tumor growth in a relatively short period of time. And it's been certainly tested in animal models that if you feed a diet high in iron, you get accelerated tumor growth. Just to and I'll talk a little bit more about this connection and show you some papers that establish this connection. And so it's known that iron homeostasis is regulated differentially in cancer cells, but there's actually surprisingly little known about how that happens. So let me just outline the goal in the project we have, which is it's funded by a grant from the National Cancer Institute. The idea for us is we would like to elucidate the mechanisms that lead to the changes in the iron metabolism network. And one reason why that's not so straightforward is because I'll show you in a minute that actually this network is a very, very well connected network that has a number of feedback loops in it. And so if you jiggle proteins at one end, it leads to all kinds of changes throughout the network. So if you want to understand the particular pattern of changes, you really need to understand how those all are connected to each other. So the basic outline is the following. Basically, characterize the two phenotypes. So do a bunch of measurements, measure proteins that are involved, measure metabolites that are involved, to just have a basic inventory of what the two phenotypes look like. Then you build a computer model of a normal cell. Okay, so it's a model that I will calibrate to the phenotype of a normal cell. So then this model will not fit the malignant phenotype. Okay, so then what I can do is I can simply use this computer model as an experiment. As an experimental system, and I can tinker with the model. So how could I change the model to generate the malignant phenotype? Okay, and it's very easy to do in a computer. I can upregulate proteins, I can cut connections between proteins, I can do any number of things. I can then simulate and see what happens, and what sort of changes fit what I see in a cancer cell. So I might find maybe, 6 different ways in which I could do this. I could, for example, add some external node pretending I have some oncogene that regulates one of these proteins differentially. I can do any number of things, and then I can decide how to prioritize these and go into the laboratory and in a very targeted way pursue these hypotheses. So, and that's really, in my mind, that is the fundamental function of mathematical and computer models. They allow you to do experiments in silico, if you like, and save yourself some time and money in doing experiments in the lab. It's yet another tool to help you design experiments in the most efficient and most effective way. Okay. So we're doing this in cell culture. The cell culture is there are several cell lines. They're all derived from primary human mammary epithelial cells, and these cell lines are then successively mutated. You introduce several mutations, the most important one being a mutation of the RAS gene, and each successive mutation has acquired yet another skill that a cell needs to become a malignant cell. So cells need to acquire a number of different skills to become cancerous, and this series of cell lines captures that. And these were initially done by the Weinberg lab at MIT So Weinberg is one of the authors in the famous Hallmarks of Cancer article that was published in 2000, where precisely these different skills were spelled out that cancer cells need to acquire. So that's the model system. Our collaborator, Susie Torti at Wake Forest, has a lot of experience with this system, and it's been excellent to work with. So let me just tell you a little bit about iron metabolism, intracellular iron metabolism, the very basics. The way you want to think of it is essentially like a temperature control in your house. Right, so the temperature corresponds to the level of labile iron in the cell. So that's iron that's available for reactions. And so that's like the temperature. So you have two things to regulate your temperature. You have a heater, right? So here's the temperature, labile iron pool. Then in your house you have a heater that increases the amount of heat you have. So those are two import proteins, transferrin receptor and DMT. DMT1 in breast epithelial cells, this is not expressed. It's only the transferrin receptor. So what this does is it takes iron that's bound to transferrin in the circulation and brings it into the cell. And you have an air conditioner that reduces the temperature. And in this case, there are two such functions. There's iron export, and that is done by a protein called ferroportin, which exports iron. And then there is iron storage, ferritin. So ferritin is a protein that can bind as many as 4,000 iron atoms and store them. And of course, for this to work, your house needs to have a thermostat. And this thermostat is actually not in this picture. So those are the two proteins called the iron regulatory proteins, and they essentially sense iron levels, and in response regulate these different features. So that's in essence what sort of the core of iron homeostasis regulation looks like, this control system here. So here's a picture, right? So I should say maybe this is what corresponds to the left-hand panel in this three-panel picture I showed you, right? That's the analog of sort of a cartoon level depiction of what goes on. And so let me show you what corresponds to the middle panel. Okay, so the middle panel. So here you have the cell. So this is a cell designer drawn picture, which you probably recognize. So here's the labile iron pool. Here's the transferrin receptor. So it brings in iron, so it increases the labile iron pool. So there's a positive regulatory effect. Here's ferroportin, the iron export protein. So this decreases the labile iron pool. Here's the storage protein. That also decreases the labile iron pool. Here are the iron regulatory proteins, so that's the thermostat, right? As the iron levels go up, it doesn't downregulate these proteins. Basically, it affects their ability to bind to mRNA, and the effect is that it will. So if the labile iron pool goes up, then these proteins are less able to regulate these other proteins, which then leads to an increase in, leads to a decrease in how much transferrin receptor copies are there and bring iron in, and it leads to an increase in these two. So as this goes up, this goes up, this goes up, and this goes down. Right? And so you can see here you have these three feedback loops. Here's one, so that's a negative feedback loop. Here is another negative feedback loop, and here is a third negative feedback loop. So from the point of view of, as a mathematician, you would say if you have three negative feedback loops like this, you can realize just about any dynamics you want. It's a quite complicated system, even though it looks sort of relatively simple here. So this is maybe the analog of the middle picture. So let me, before we go on to the right-hand part, let me just go back to, now that we've introduced the proteins, let me just go back to talking a little bit about the connection between these and breast cancer. So here you have a paper going as far back as 1980 that talks about transferrin and transferrin receptors and the relationship to breast cancer. Similarly, in a paper more recently, 1999, c-Myc regulating ferritin and iron regulatory proteins. So there's a clear connection between oncogenes and iron. And here's a paper that talks quite a bit about this. If you want to get a survey, this is by our collaborators, Susie Tordi and her husband, Frank Tordi, the role of iron chelation in cancer therapy. They published another paper just a year and a half ago that focused on ferroportin. So this is the iron export protein. And what they showed with the help of a number of large microarray data sets from breast cancer patients that this iron export protein by itself represents an excellent predictor of disease outcome. So you can, in women in which ferroportin is significantly downregulated, the prognosis is significantly worse than for patients in which this protein is unchanged. And so this protein by itself is a much better predictor than other gene sets for these particular data sets that have been studied quite a bit in a number of places. And so that has started a lot of focus on this particular individual protein. But of course, as you can see from here, right, to change the expression levels of this protein, I mean, I could change any one of these 3 or any one of these 4, right? And it would simply, through these feedback loops, the effect might propagate to ferroportin and downregulate it without anybody ever doing anything to that particular protein itself. That's why it's useful to understand the whole network and the way effects propagate through this network. So one of the things we did, actually as part of the cohort in the REU after the year Ali was there, we built a differential equations model of this network here. So this is the right-hand panel in this 3-panel picture. Okay, so I'm just showing you these equations, and there's no need to really look at them in any detail. This is transferrin receptor, and it changes with respect to as IRPs change. Here's a degradation term. So actually, if you look at these carefully, it's not all that much of a problem. But in any case, so this is part of a paper which is going to appear in just a little bit in Journal of Theoretical Biology. It's available online already, I think. And so now that allows you to create the kinds of pictures you see on the right-hand side in this panel, actual changes in concentration. The problem is that, so this system has 5 equations in it, the whole one. I'm just showing you 2. So you have five nodes in this network. There are 5 equations, and these 5 equations have 18 parameters in them. And again, you can find values for these parameters in the literature, but they will be from a different cell type in a different condition. And so it's not so clear whether one actually wants to include these parameters. Now, there's more to iron homeostasis than this small network. Right? There's a lot more to iron homeostasis. Oh, I see. I'm sorry. I'm getting ahead of myself. Let me just say a little bit about this model that we wrote. So you can go and you analyze this model, and of course, if it's going to be any good, you need to validate it in some way. So we've done that. So one of the things, and remember I told you before that just given the connection between these different feedback loops, you can essentially produce any type of dynamics that you want if you just put the right kinds of functions on these nodes. Now, for the particular equations that we have, it turns out that there is no oscillatory behavior, and that is true for any parameter, any choice of these 18 parameters that you might choose. Right, so it's independent of parameter choices. You get no oscillations. You get a unique stable steady state. And again, independent of parameter choices. Now, why is that important? So the reason we looked at this network in the first place, this small one, is that our guess is that this network probably is not changed at all in cancer cells. It stays the same. It is simply regulated differentially from the outside. And so the first thing to understand is what is the dynamic behavior of that network by itself if you just leave it alone. And that's exactly what we've done in this paper, right? If you leave it alone, it does exactly what it should do. You would expect iron to be regulated very tightly, right? Because there's all kinds of problems with reactive oxygen species, and so you don't want to have iron going, fluctuating wildly, for example. And that's exactly what this model does. And it fits some experimental work we've done. It's an iron challenge, for example, and one particular observation that if you overexpress ferroportin, right? I told you a minute ago that what is bad from the point of view of prognosis is that if this is underexpressed. But if you overexpress it, then ferritin expression is reduced, right? The storage protein. And again, it's independent of parameter choices, which is important because we cannot parameterize this model. So if it's going to be any good, it better be independent of what parameters you choose in this particular case. So that what drives the dynamics of this is a lot just the topology of the network and the particular kind of reactions that are put on there. Now, as I said, there's much more to iron regulation than the core network that we put down. So this is a network reconstruction we did of the iron metabolic network in 2009. So this is the network based on the literature at the time. It has about 150 molecular species in it. And you can see different compartments. What we did in this paper is we looked at the differences in four different cell types. So hepatocytes, macrophages, etc. And by now this picture would be much more complicated if we did it. One of the things you see here, this note here, you can't read it, but it says ROS. So this is the whole reactive oxygen species response network, which one we really need to, which is very important, and it really needs to be blown up. So initially we ideally said, well, we will just model both of these networks together until we did one. And so ultimately the goal is, of course, to make a dynamic model of the sort that I showed you for the much larger network. And one of the projects we have at the moment is sort of a bioinformatics project that goes with it. With this, we have a number of microarrays of the cell lines that I talked about, and what we would like to find out is, right, that what's nice about these cell lines is that you know the specific mutations that have been introduced to make these cells malignant. Right? And so what we're looking for by analyzing the gene expression patterns is to see if I find what all the downstream effects are of these mutations, how do all these downstream effects intersect with this network? So where are potential connections between this? Which part of the network could be perturbed? And I thought that was me. So that's one of the projects that comes with this, and so we've gotten a start on this. We've expanded our network. For example, we've separated out the iron regulatory proteins. We have included the heme system, heme degradation and synthesis. One of the important molecules that comes in is hepcidin, which has been known quite a bit as a systemic regulator, but it actually plays a role in intracellular regulation as well. So this is the next step, and I should say that's what last summer's REU program has done. So we're making use of the free labor of all these smart undergraduates that come in. And so the problem is that once you make these differential equations models that I showed you earlier, once you make those bigger, of course you introduce more and more parameters, right? And then what you have is you don't have a quantitative model, right? That's the good thing about differential equations, they're quantitative. But if you can't get the parameters right, then you don't have a quantitative model, you have a qualitative model. And so we said, well, let's just build a qualitative model in the first place. And let's ideally build a model that fits many of the data we have. So we started doing this with Western blot data. Okay, so now we're going to get mass spec data pretty soon, but these are Western blot data, so these are not quantitative, right? So you have qualitative values or semi-quantitative at best. So you may as well simplify things, and rather than having real numbers as concentrations, let's get categorical values, right? 0, 1, 2, low, moderate, high, or low and inside the nucleus and low and outside the nucleus, whatever you need, but discrete categories, right? So in this case, if I then look at a state of the system, right? I list all the proteins in the system, all the molecules in the system, and I can describe them, right? So labile iron pool is moderate. Transferrin receptor is expressed highly. Ferroportin and ferritin are very low, and so on. That's how you would read a state like this. And in my experience, that corresponds a lot with how experimentalists think about their data. Right? This gene is overexpressed. Maybe you can say it's overexpressed 3-fold or 2-fold or 10-fold, but you will not probably think about it as being overexpressed 2.785-fold. And so how do I build a model, right? How do I get dynamics into this? Well, so as an example, right? Let me look at heme. ALAS1 and HO1. So ALAS1 is regulated by heme, and of course it has self-regulatory property. And so I can simply take a table like this, what I would call a transition table, right? So to look at what ALAS1 at the next time step would look like, I know what heme looks like at the current time. I know what ALAS1 looks like at the current time, right? And then I can determine what happens to ALAS1 in the future. And how do I do that? Well, it's biological information, right? If I know that heme is moderate and ALAS1 is high, then ALAS1 will, at the next time step, will be moderate because you have a negative regulation from heme to ALAS1. So that's what this table expresses. So this is biological information that you use to fill in this column. Okay, so this is best done by an experimentalist, not by a modeler. Okay, I mean you could, right? So if you wanted to do this in software, right, you wouldn't want 0s, 1s, and 2s here, but you actually would want to have text there, like moderate, so you don't need to remember what all this stuff means. Okay, but so I do this for every node, and I get all these transition tables, and then if I have a particular network state, I can compute the next state by applying these transition tables. So I'm describing to you a different way of modeling, right? If you don't like differential equations, or if you think it's not the right kind of thing, this is a different way of modeling. It's a qualitative way of modeling. But what the advantage of it is really one step removed of what you find in a paper that an experimentalist will write. In fact, we're sort of experimenting with this. We have another cancer project on melanoma, and so there our role is a human interface. We would like to understand what the right interface is. And so we get somebody to essentially input their biological information about a system by filling in these tables. And so you get a discrete model. It has all kinds of nice features. Let me not expound on those. You can draw output from Excel spreadsheets that's quite similar to output you might see for your data. So maybe it looks a little bit different, but you can see here you have a number of essentially you have all of the molecules in this expanded model that I showed you. Here you have time, so that's time 0, time 1, time 2, time 3, and so on. And here you have concentration, which now is 0, 1, 2, rather than some real number. And you can see how each of these molecules evolves, right? So this is actually a limit cycle, so you have periodic behavior of each of these. This one here is constant, it doesn't change at all. Same with this one. Here you have, again, periodic behavior. So you can visualize the output from these models in ways similar to what you're used to, right? Maybe this is not the best way of doing it, but we haven't put a whole lot of effort into that part. So what's the next step, right? So now we have this model, and it's an expanded model from the differential equations model that I showed you. You can check that qualitatively they actually behave. They give you the same information, unique, stable, steady state, etc., that I showed you before. And so now we can start doing something. And what we would like to do, as I told you, is we want to use the model which is built for normal cells. How can I tinker with the model and change it so that I get the cancer phenotype? So in this case, the cancer phenotype is here, right? What we observe, what has been observed is that ferroportin is lower, transferrin in the particular cell line that we're looking at, transferrin receptor is up, ferritin is down, the labile iron pool is up. And what we can do with this model is we've identified one protein, and at this point I don't want to tell you which one it is, but we've identified one protein that if you upregulate that, and you can implement this in the model very nicely, basically you increment the values in your transition tables. If you upregulate that one protein, it produces exactly the cancer phenotype. So now we're in the lab upregulating that protein to see what happens. And we're pretty confident that that's going to work very nicely. What's good about this particular model type is precisely that it still works well if you expand this network to have 100 nodes. Right? You're going to have to spend a fair amount of time filling in these tables. And some of these tables get quite big, but there are a number of ways in which you can implement sort of autofill features, right? That you fill in some key changes, and you can then deduce a number of other changes that will have to happen, so that there's relatively little that you need to do. So in addition to trying to understand breast cancer and iron, we also are interested in building bioinformatics tools for experimentalists. Basically, what I would like to do is develop software that makes me completely superfluous in using mathematical models as tools for discovering new biology, right? Because where do things need to go, right? We need to, everybody agrees by and large that quantitative methods have to be introduced as a matter of course into biology, right? The field of computational biology will have to go away, right? There will be no more computational biology, no more than there is computational engineering. Right? It is computational by nature, and biology, or at least large parts of it, will become computational by nature, and so you don't need the adjective computational anymore. Right? I think in large part it will go there. But if the only way you can do that is by bringing together a modeler and an experimentalist, right? There's many more experimentalists than there are modelers, and many more biologists than mathematicians or computer scientists. So it simply does not scale. That's a model that doesn't scale. And so what you need is you need software tools that enable a biologist without getting a master's degree in math to do mathematical modeling and have that as a tool in their lab, right? And ultimately, especially when it comes to design of experiments, you should spend a lot more time on a computer before you ever set foot in a lab. In fact, I just heard a talk a little while ago of somebody who said that laboratories will become peripherals to computers. So I wouldn't go quite that far. But if I was a biologist, I would find this very frightening. Okay, so you need software tools, and just in the last minute or two that I have, let me just go through those, right? So the first thing you need is an interface that is better than the interfaces current modeling tools have. And you need a good framework to analyze models, right? So if you have differential equations models, the two things you do is you do parameter estimation. So you have a model that has some unknown parameters in them, and you fit them to experimental data that you have. Okay. And another thing that you need to do is you need to be able to do what's called bifurcation analysis. That is, if I change a parameter, you know, how does that affect the steady state behavior of a model, for example? Do you go from having one steady state to having a bistable system, for example? How does that depend on the parameters? So those are all things that you need to know. And so we've developed some software for the kind of discrete models that I showed you. So here's one, and let me just point out that, so this appeared last year in BMC Bioinformatics. Again, it has 3 of the authors on here were RU students. And here's another paper. So this does the analog of parameter estimation for discrete models. And we're now in the process of sort of turning this into one big software package. So if you're interested, I can certainly tell you more about it later, but I'm out of time, so let me just move on to the future. And you know, borrowing a political slogan from the 1930s, a chicken in every pot, meaning the kind of modeling software I was talking about before in every laboratory, right? That has intuitive interfaces that you can build these models, right? Filling in these kinds of tables and doing a number of easily carried out tasks that allow you to perform experiments in a computer before you go into your lab. So this is just a summary of what I talked about, right? What you want to use these kinds of dynamic models for is essentially you build them, and which kind of model you build depends on what kind of question you have to answer and what kind of data you have and what kind of extra information you have. For example, if you want to do metabolic engineering and you want to optimize E. coli to do a particular metabolic process 5% more efficient than it's doing it, then the kinds of qualitative models I showed you will not do you any good. You need something different. But so you build your model, you validate it once you trust it, you use it as a tool to generate hypotheses. You go in the lab, and it will save you time and it will save you money. So let me just tell you people that are involved in this. So these are the folks from Wake Forest, Steve Ackman, Frank and Susie Torrey, that have been working with us on the iron metabolism part. Here's a long list of people who have worked in my lab, who have been collaborators. Greg was a postdoc, and I have in italics, I have all the undergraduates. So these were all undergraduates that over the years have contributed to this project in different ways, and it's been great. So they've become an important part of my lab. and we've had funding from different sources for different parts of this. The NIH is funding the breast cancer work, and the NSF, actually a grant from from the engineering directorate, is funding some of the theoretical work that goes into it, which I have not talked about at all here, and Department of Defense that has funded some of the software development. So thank you very much.
Is the information in the transition table more or less equivalent to the information in the regulatory diagram? Or do you still need some intuition from the experimentalists?
No, it's not. The two, in some sense, are independent. I can, if I have the wiring diagram, the regulatory diagram, depending on how I fill in my transition tables, I get vastly different dynamics. So you need both. Right? Because and again, as I said, if you just look at these feedback loops, it's known that you can generate all kinds of different dynamics from that, depending on how you fill in the transition tables. Yes?
So one thing that sort of makes cancer very complex is the cooperation between cancer cells and normal cells and other cells in the body, like macrophages and cancer cells. And there, macrophages make proteins that are involved in iron metabolism. So I noticed that you are starting to separate some of the metabolic events between cell types. What about the modeling of the interaction between cell types?
Yeah, so that's so our aim is to go multi-scale, right? That's the buzzword today. If you want to get funded for sort of method development modeling, you have to do multi-scale. You have to do stochastic and multi-scale. Those are the two things, right? And so the next level would be a tissue scale model, and it's very interesting. So, for example, it's known that if you grow this type of cell in three-dimensional cell cultures rather than as a monolayer, you get very different expression patterns. In particular, in the iron regulatory proteins, and then if you include a different cell type into this, such as macrophages, right, then you get very interesting interactions. But it's actually already at that level. It's quite complex to build a model, and then of course, if you go to a larger model. But you're absolutely right. That is the place to go, right? Because it's certainly understood that the environment in which a cancer cell and the environment in which a tumor lives is crucial in determining what happens.
Repeat the question.
Oh, I'm sorry. Yes, I should repeat the question.
Can you use your T3 model to estimate your steady state?
So the question is, can we use this model to estimate the steady state? So I showed you two types of model. The differential equations model, to find a steady state, basically I take the equations and I set them equal to 0. Right? That's computing the zeros of the derivative. And what you end up with is you end up with a system of equations. They happen to be polynomial equations. You can analytically solve those, and you can find the steady states. For the other discrete model that I showed you, this one is small enough. I can simply exhaustively simulate this model, and see that there is a unique steady state. Or if the model is larger, I can essentially do the same thing. It turns out even in this discrete case, I can represent the transition tables by polynomials. They're sort of weird polynomials, but they're polynomials, and I end up sort of solving a system of equations to find the steady state. So in both cases, the answer is yes. Yes, sir.
So one of the problems that people doing computational work face is the interaction with experimentalists who simply do not trust. And it's not only bio field. I mean, people in mechanical engineering materials have exactly the same issues. So one of the reasons is that parameters estimation, noise in the data, and so on and so on. So I wonder, what is your comment on this? I mean, Is there any way to convince this community to actually use the tools which we are developing and help to improve them actually?
Yeah, so the question is how do you address the fact that experimentalists typically don't trust computational methods? So I would say my comment is that anybody who believes microarray data, right, after you see what Affymetrix does to pre-process the data before you ever see them, I think that person has no leg to stand on when they question your methods. Yes?
Suppose you have 20, agents. Would you need an astronomical number of experiments in order to fill in this table accurately?
Mm-hmm. Yeah, so that's right. So here's a question: how do you get all the information to fill in these tables? So if you have a larger model, you have a lot of things to fill in. So two things happen. There are many constellations in these tables that are biologically not feasible. Okay, because if you have 20 different proteins in the cell and only one of them is expressed, right, that will never happen. So a lot of these states are actually infeasible states, and you can cut down the tables. But still, you will be left probably with a lot of them that you don't know. And so that's where this capability of parameter estimation comes in, right? You should be able to say, well, I don't know this, so instead of filling in a 0, 1, or a 2, I fill in a question mark. But I have this set of experimental data, right, that might help. So what I can do is I can calibrate this model by finding specific values for these question marks so that the model at least coincides with what my data do, right? And that's what you do with differential equations or any other kinds of computer models. You calibrate them to data, and there's typically a number of different choices of parameters that will give you the same type of behavior. So that's the two things I would say. There's fewer things you need to fill in, and it would be expected that some of those you don't know what they are. Yes. Actually, because one of the things you might need to take into account is where. So the question is when is high, medium, low not enough? So how do you determine the number of discrete states that you need to have in your model? So one of the things you might have to include is localization in the cell. Is a protein in the cytoplasm? Is it in the nucleus? Is it bound to a particular other protein, or is it unbound? So you might easily end up with 5, 6, or 7 different states. Yes. Yeah, so the question is, as you make this model bigger, how do you verify that you still don't have oscillations in it? Well, I mean, essentially you do it the same way, right? As we did here in the differential equations case, once models get really big, what you probably need to do is you need to do extensive simulations, right? So you choose lots of different parameter values, you simulate, you see that you get a steady state rather than oscillatory behavior. So that's that's not as good as an analytical study of this, but analytical studies typically are not possible. With a discrete model, so if the model is small enough, you can typically do an exhaustive simulation of it. So if you have as many as, say, 30 different molecules in your model, you still might be able to exhaustively simulate. Beyond that, so we have tools from actually computer algebra that reduce these questions to solving systems of equations that will extend that capability to models that are maybe of size 50 or 60. Once they get too big, then you just can't do it. And there too, you need to do simulations and be happy with nothing that's exhaustive.
We have time for one more question.
How many days end up with marriage?
Well, you know, so in my experience, to me, finding good collaborations where people trust each other has been by far the hardest thing to do, and I've wasted the most time and money on the unsuccessful ones, and they were unsuccessful for many different reasons. It's just not a good match. Your goals are different. all kinds of things, right? And it's no different, right? You're asking, if I come to you and I say, you know, do this experiment for me, it's going to take 2 weeks of one of your postdocs, right? You would be reluctant, just like you come to me and you dump a bunch of data on my desk and say, tell me if there's anything interesting in these data. You know, spend the next 2 weeks staring at them.
And then actually it's a lot of talk.
Yeah.
I'm sure you'll say the same thing about the people we were collaborating with, talking communications.
There's a huge amount of overhead involved, of time that you need to invest and talk to people, right? And I ask the same stupid question 5 times over, you know, and I would still like to get an answer rather than saying, you know, go away. Yeah, I agree, you're absolutely right.
All right, I wanted to thank you all for attending our first seminar, and also to invite you. Right now, we are having a little reception just out in the atrium, I believe. And then